Welcome back to the Deep Dive. Today we're tackling a big one. We've got a stack of expert reports, academic findings, basically all the dense technical stuff. And we're going to distill it all down into a fast, fact-based review of the pandemic and really the RNA vaccine rollout.
And our mission here is really about scientific clarity. We're trying to sidestep all the noise, the political narratives, and just synthesize the documented facts, the tech, the manufacturing, the regulation. So if you want to get scientifically up to speed on this, this is the deep dive for you.
Right. This isn't about criticizing past decisions. It's about looking at where the science met, reality, and what we can learn from that.
Because getting the science right now means the next time any future medical response can be built on much, much firmer ground.
Okay, so let's unpack this. I think we have to start at the very beginning. The origin of the virus itself.
Right.
The official position, from governments, international bodies, is natural occurrence.
That is the official public line, yes. But what's really striking in the sources is this strong alternative hypothesis that has really solidified in research circles the possibility of a leak from the Wuhan Institute of Virology. And this whole idea is tied to a specific kind of research called gain of function, GO.
Right, GOF.
Yeah.
That's where scientists genetically modify a virus to make it more infectious or more dangerous. Why would anyone do that?
Well, the stated goal, the academic goal, is to see how a virus could mutate in the wild.
To get ahead of it.
To get ahead of it, theoretically.
Yeah.
To anticipate a pandemic before it happens. But as all the sources stress, there's this huge inherent risk. The potential for weaponization, or in this case, just an accidental release.
And here's a fact that I think is crucial, and it's documented. The U.S. National Institutes of Health and the Department of Defense were actually funding some of this research.
They were.
At the Wuhan Institute, so a direct U.S.-China collaboration.
Precisely. And that's where academic curiosity meets this massive biosecurity risk, right? It immediately brings up the question of, how do you separate academic goals from these potential biosecurity conflicts?
That's a huge question. So what's the actual technical evidence that points to the leak idea over the natural one?
It really comes down to the genetics. When scientists analyze the virus's sequence, they found certain mutations that are, let's say, unlikely to occur in nature.
So a kind of genetic fingerprint.
A kind of fingerprint, exactly. And that signature is what has made the leak hypothesis the kind of prevalent view among many, many scientists. It's a compelling argument, just based on the genomic.
Okay, so that sets the stage for the response. The virus is spreading like wildfire. It's an RNA virus, so it mutates fast. And so the traditional inactivated vaccines were just seen as too slow.
That's the key rationale. Necessity really drove the choice of RNA vaccine technology. The concept itself it's quite elegant. You introduce some messenger RNA for the spike protein, wrap it in a little protective shell, a lipid nanoparticle, an LNP. It gets in your cells and teaches your immune system what to fight. It was fast.
But that speed came with a cost.
A huge cost in terms of manufacturing maturity.
Okay, this is where, for me, the story gets really interesting. We're moving from the clean science to the messy reality of the factory floor. The issue wasn't the idea, it was the RNA purity.
Yes.
I never even thought about the purity of RNA. Can you walk us through why it's so hard to make it clean?
Sure. Think of it like baking a really long, complicated cake. That's your long-chain RNA. You're using an enzyme to link all the ingredients, the nucleotides, together.
Okay.
But when you do something that complex that quickly, you just inevitably get a lot of junk. Burnt crumbs, incomplete bits, those are the byproducts, DNA fragments, double-stranded RNA that you didn't intend to make.
And I'm guessing these byproducts look a lot like the actual RNA you want, so they're hard to separate?
Exactly. The tech to perfectly purify that long-chain RNA from all that related junk is incredibly complex. And frankly, it was still in its infancy when these vaccines were deployed.
This is the part of the sources that it floored me. What were the documented purity figures?
This is from experts like Professor Hiroshi Abe at Nagoya University. He reported that the initial clinical trial batches,
The best case scenario.
they had a purity of about 78%.
78. Wow. That already sounds low for something going into billions of arms.
It gets much, much lower. The shocking fact is that the commercial batches, the ones that went out to everyone, had purity levels recorded as low as 55%.
55. Wait.
Yeah.
55. That means 45% of what's in the vial is manufacturing byproducts.
Correct. DNA fragments, unintended RNA structures.
Help me put that in context.
Yeah.
How does that compare to a normal drug?
The contrast is It's immense. For almost any generic drug you take, like a painkiller, the purity of the active ingredient is typically 99.99%.
99.99.
And we were injecting a product where almost half the contents were these unpurified fragments.
And with a traditional drug, if you have even a few percent of impurities, you have to do toxicity testing on each one. Right.
That's the protocol, because the impurity itself could be toxic or cause inflammation. But with RNA vaccines being so new, those regulatory standards they didn't exist for these specific impurities.
So we basically injected something where the effects of 45% of the contents weren't fully verified.
Before massive global distribution, yes.
This completely changes the context of those liability waivers.
Yeah.
I mean, when this 55% number came out in academic circles, a lot of doctors said they wouldn't have taken it or promoted it.
It totally changes the risk calculation. And if you connect the dots,
The waivers.
this low purity standard is almost certainly the reason the pharmaceutical companies insisted on those liability waivers.
So you're saying the waiver wasn't just, standard corporate practice. It was an insurance policy against a known documented technical risk, a manufacturing standard that was way below the norm.
That is the compelling conclusion when you put the technical facts next to the legal outcomes. They knew the technology had this shortfall at the time.
Wow. Okay. Let's shift focus to the delivery system, the lipid nanoparticle, the LNP, the little shield for the RNA.
Right. So the LNP structure, that dictates stability and critically where the vaccine actually goes in your body.
And the initial tech had problems.
Two major ones. First was stability. The early LNPs were very fragile, very heat sensitive. They needed that ultra cold -80-degree storage.
Which is just a logistical nightmare. Imagine trying to manage that in a school gym somewhere. Failures must have happened.
They were hard to avoid, and if the temperature control failed, the vaccine could become inactivated, basically dead.
What does that mean if you get injected with a dead vaccine? Is it just that it doesn't work, or could the breakdown products be harmful?
And that's the huge blind spot.
Mm-hmm.
The link between injecting an inactivated vaccine or its breakdown products and adverse events is largely unknown. It wasn't studied properly.
Okay, and the second LNP problem?
Systemic distribution. The claim early on was that it would stay local, in your arm.
I remember that.
That was quickly proven false. We confirmed the vaccine circulates throughout the entire body. It can get to vital organs, the brain, the heart. And the safety evaluation for that systemic spread is still highly questionable in the literature.
But the science did evolve fast. The sources say that newer refrigerator-stable LNPs have been developed since.
Which is the silver lining for sure. It shows the tech is moving toward a safer, simpler model for the future.
But we have to talk about the human cost and the social side of those initial safety issues.
Definitely.
The reports are clear that adverse events were stronger in younger people. It even prompted government advisors like Omi Shigeru in Japan to say inoculation should have been handled more cautiously for them.
And critically, the official reporting systems for adverse events are widely believed to be a massive underestimate.
Because only the really severe cases get reported.
Exactly. Only severe or long-term impacts tend to get captured. So the true incidence rate is almost certainly higher.
On the technical side, do we know what actually drives how severe an adverse event is?
Yes. It's the variability in how much spike protein gets produced. With this RNA technology, it's very hard to control exactly how much spike protein each of your cells makes, and that uncontrolled variation is directly linked to the severity of the reaction.
Which brings us to this fascinating social issue the reports documented: researcher silence. The molecular biologists, the ones who knew about the 55% purity problem, why did they stay quiet?
The sources suggest a lot of it was fear. Fear of jeopardizing other sensitive, high-profile science projects.
Like what?
The big one mentioned is the construction of a BSL-4 lab in Nagasaki. That's a maximum containment facility for things like Ebola. Researchers were worried that speaking out about the vaccine's flaws could tank the funding or approval for that lab.
So they prioritized their long-term projects over disclosing short-term problems in an emergency.
It appears so, which, ties this whole conversation right back to where we started.
Gain-of-function research.
It closes the loop. The material is unambiguous on this. No matter your containment level P2, P3, P4 accidents happen, it's just a fact. We have precedents, like nuclear accidents, that confirm that failures and leaks can and do occur.
So researchers have to anticipate that. They can't just wish it away.
They have an ethical mandate to be prepared. The sources argue that if you create a high-risk artificial virus, you must have isolation centers, treatment protocols, medical staff all ready to go before you even open the lab. An academic reason isn't enough if the risk is existential.
So bringing this all home for our listeners, the COVID RNA vaccine rollout was really an emergency deployment of a new technology. It was fast, which was necessary.
But it ran headfirst into huge scientific and manufacturing challenges: purity, logistics, data transparency.
And the key takeaway on its value.
I think we have to be very careful not to equate this with traditional vaccines. This was an expensive, multi-dose vaccine with limited effect and serious purity issues. It's not in the same category as the low-cost, incredibly effective, stable vaccines for things like polio or measles.
So what's the single biggest lesson for next time?
The expertise was too narrow. In the media, we mainly heard from clinicians and epidemiologists. The crucial voices from drug manufacturing, from R&D, the people who understood the 55% problem, they were largely missing from the conversation.
We need a multidisciplinary review.
Absolutely. And that is, thankfully, now underway. Formal guidelines for mRNA pharmaceuticals are finally being created.
Now, we have to use this experience to fix our institutional design for the next crisis while it's all still fresh.
Which leads to our final provocative thought for you. Given that we know adverse events were underreported, the only way to get a true picture of long-term safety and effectiveness is through massive, comprehensive data collection.
Across the whole population.
Across the entire population. So it raises a really important question. Should governments be leveraging national ID systems, something like Japan's My Number system, to conduct the huge long-term studies we need to protect public health in the future? Something to think about as we navigate this post-pandemic world.